for this meeting. And also I will say something regarding assessment criteria before I start talking about the first lecture. Let me share my presentation right now. So my name is Liana Ramishvili, and so I will help you to study cell biology. So as I told you, the main topics of our lecture is regarding the basic types of the cells. We must remember which type of the cells we have, what are similarities and what are differences between the cells, what are the main characteristics of the cells, but also I want to tell you about the basic literature that we must use during our course, especially this is the first one, Gerald Karp's Cell and Molecular Biology. And also I will share this online book on Google Drive, especially, so I will provide the link for this. Also, I will give you my email, my university email, of course, and anytime you can contact me regarding with the help of this mail if you have some problem regarding the to somehow if it's not available still, I mean this online book, you can contact me and I will share it this book once again. So these are the basic
اسمعي خليكي اليمين حتى توصلي عند إشارة تمام؟ يعني خليكي على اليمين حتى تخلصي اليمين حتى تخلصي هذا الشارع تمام؟
I can't understand you, my dear.
يلا.
And I want to ask you, in case you you want to say something, yes. Only in this case, please turn on your microphone. Yes. During the lecture, please provide that. Please ensure that your microphones won't be turned on. Yes. Turn off your microphones during the lectures because we will have some kind of noises. Yes. To avoid these noises, please turn off your microphones. But if you have some question, of course, only in this case you can turn it on. Yes. Do you have some questions? For example, regarding the material or regarding, and I want to ask you, or everyone has a Moodle, yes? Its own Moodle with the help of university mails, of course, yes?
Yes, doctor.
So this Moodle site is available for you, yes? I want to ask, is there any technical problems regarding the Moodle?
But if we want to contact with you, we will gonna chat in the email.
So yes, you must use your university mail because your official email is your university mail, and everything that I want to tell you, for example, yes, I will contact with you with the help of this mail because I have only this mail that is indicated in Moodle system.
Understood. Thank you.
Yes, you're welcome. You're welcome. So regarding the assessment criteria, I want to tell you shortly, of course, the very important component of our assessment is a seminar activity. I hope you already know about it. So nearly 20 scores go to seminar activities. So you have 10 attempts, yes, 10 chances to collect these 20 scores. Nearly we have 13 or 14 seminars, yes, during the semester. Overall, we have 15, of course, but today you don't have any seminar activity, of course, yes, because today is the first lecture, our first meeting. Starting from the next Monday, yes, you can start your seminar activity, and every student has a maximum 10 chances. Yes, you must participate in 10 seminars to collect these 20 scores, and participation in one seminar will be maximum 2 scores. Yes, so if you will participate in 10 seminars and you will get 2 scores on each of this participation, you will collect these 20 scores, of course. Yes. Another component is a midterm, middle exam, of course. This is also 20 scores in the middle of the semester. The next component is a quiz. We will have 2 quizzes, 5 scores each. Yes, overall 10 scores goes to a quiz component, and also last one is a presentation. Yes, but presentation we will do it at the end of the semester. Yes, very last last meeting at the end of the semester will be devoted to these presentations. So you must prepare one presentation, and you will choose some topics that I will send you, of course, from cell biology. You will choose one of the topics and you will prepare this presentation, and you will get 10 scores, maximum 10 scores in your presentation component. Yes, so these are the main components that we must fulfill during the semester, and of course finally you will have the final exam, of course, 40 scores. Yes, at the end of the semester. So I think everything is clear now. What kind of activities we're going to have during semester. So let's start our live lecture now. If there's no questions regarding assessment criteria, for example, we can start our lecture. So as I told you, main topic is the basic properties of cells and basic types of the cell. But before we start talking about the characteristics of cell, let's remember about the scientists who discovered cells, for example, and who contributed to a formulation of cell theory. Yes, so-called cell theory. Of course, first scientist that we must mention today, this is Robert Hooke. Yes, you remember Robert Hooke. He examined cork slices, and he really introduced, he was the first scientist who introduced this term cells. And we can also see this picture depicting this situation, depicting the image that he saw under microscope, these chamber-like structures he called cells. And so he was the first scientist who introduced this term, of course. After Robert Hooke, we must also say something about this second scientist, another scientist, Anton van Leeuwenhoek. Don't mix it up. Yes, this is Robert Hooke, and this is Anton van Leeuwenhoek. Yes, another scientist who discovered actually bacterial world, and also protists, one cellular organism. So what we can say regarding Anton van Leeuwenhoek, he discovered a microscopic world because no one could see them, yes, before, and he called this organism animalcules. Yes, very strange term, but he called so this protists when he was examining the pond water. Also very important thing is that he created first known microscope. Also we must say something regarding this, because in that period everyone was trying to create some kind of microscope. So he was one of the scientists who created this first known microscope. And as I told you, another discovery is regarding bacteria. Yes, bacterial world, when he was examining scrapings from his teeth. So after these two scientists, there were also two German scientists, Matthias Schleiden and Theodor Schwann, who collected all this information regarding the cells, and they formulated actually two tenets of cell theory. What are these tenets? First one is that all organisms are composed of one or more cells, And the second one is that cell is the structural unit of life. Yes, it means that all organisms contain a cell. Yes, as a structural unit. Yes, so no life without a cell. Later, of course, when scientists discovered viruses, this was some kind of a different thing, because you know that viruses do not contain any cell as a structural unit. So only viruses don't follow these rules. Yes, please remember, all other organisms, starting from bacteria, including humans, all other organisms also, of course, follow these two tenets, and all these organisms contain cells as a structural unit. Later, another scientist, Rudolf Virchow, formulated a third tenet of cell theory, and he stated that cells can arise only by division from a pre-existing cell, and this statement was added like a third tenet for the cell theories, because he proposed how these cells are reproduced. Yes, so he was a histologist. He was trying very hard to find out how cells are reproduced. So he proposed a third tenet for cell theory. So now, what are the basic properties of cells? What we can say, how we can describe, how we can describe or characterize the cells. Yes, first one that we can say that cells are quite highly complex and organized structures. What we mean under this? All these processes or activities that we can detect, for example, inside the cell, all these activities can be remarkably precise. Yes, at least, for example, if we take one process like DNA replication, this is synthesis of DNA molecules. Yes, so DNA replication occurs with an error rate of less than one mistake in every 10 million nucleotides incorporated. So, and most of these mistakes, these errors, are corrected by repair mechanisms. Yes, so that's why we say that this process can be remarkably precise. And also we have some kind of organization. Yes, inside the cell we can observe complexity and organization at different levels. Yes, like atoms, then molecules, polymers, then subcellular organisms and cells. Yes, also we have organelles inside the cell. These are very tiny structures that carry out some distinct functions inside the cell. You remember some organelles, I hope, like mitochondria, like endoplasmic reticulum, like lysosomes, and all these organelles have a distinct structure and distinct function. Yes, and they are quite complex, and they have quite complex structure, especially mitochondria, for example. Yes, and so we have, that's why we say that we have complexity and different level of organization. Also very interesting is protein folding, how these proteins acquire these three-dimensional structures. Yes, because without these they can't carry out their functions. Yes, because most of these proteins have their tertiary and quaternary structures, and it means that protein folding is essential process inside that is carried out inside the cells. The next property of the cells is that every cell possesses a genetic program. Of course, it means that every cell contains some package of chromosomes, some set of chromosomes, and these chromosomes contain DNA molecules and, of course, genes. Yes, that contain information regarding the primary structure of distinct proteins, and the molecular structure of genes allow for changes in genetic information. We call it mutations. that lead to variations among individuals, and we call it biological evolution. Yes, it means that time after time there can be some mutations inside this genus that help in biological evolution, of course. The next property of cells is that every cell is capable of producing more of themselves. It means that every cell can reproduce. Yes, of course. We call it division, cell division. For example, if we take mitosis, one of the type of divisions, from mother cell we will get 2 daughter cells, and this genetic information is must be copied and divided into 2 parts in order to get distributed in 2 daughter cells, and every cell gets the copy of this information from the mother cells. The next property is that cells acquire and utilize energy. What we mean here how the cells can transform energy from one form into another form. For example, photosynthesis is a very good example for it. Yes, because in case of photosynthesis, cells are capable to acquire sunlight energy and to convert this energy into chemical energy of compound like glycogen, like glucose, for example. Yes, and also aerobic respiration is very good example. Synthesis of ATP molecules, yes, in our cells is very good examples. Another property is that every cell carry out some set of metabolic reactions. Yes, we call it cell metabolism, and for this they need special proteins that we call enzymes. Yes, because without enzymes it's impossible to carry out some metabolic reactions inside the cells. So it's closely related to the action of the specific proteins that we call enzymes, of course. And another very interesting property is that cells engage in mechanical activity. So it means that they can reveal some kind of action, yes, some kind of actions, so that we can see it physically under a microscope. For example, when we detect cell division, for example, yes, because in case of mitosis we have two tubulins, yes, some specific proteins that carry out this process. I mean distribution of chromosomes. Yes, in case of mitosis this is distribution of chromatids. They must be pulled into direction of polar sites. Yes, so and also we have other type of so-called motor proteins like myosin. You know about myosin because they are involved in muscle contraction. Yes, without myosin, no muscle contraction. Dyneins, very specific proteins that we have in flagella or cilia that ensure the movement of the structures. Yes, flagella and cilia, and kinesins also that are involved in mitosis and meiosis. So we have some kind of motor proteins, and with the help of these motor proteins, cells engage in mechanical activities. Also, cells are able to respond to stimuli. This is especially obvious in case of unicellular organisms like protists, like amoebas, for example. Yes, because we can observe how they respond to light stimuli, for example, or when we introduce some new material in the area where it lives, so we can observe this they respond to the stimuli. But in case of multicellular organisms like humans, for example, it's not so obvious, of course, like in unicellular organisms. But here we have the action of some specific proteins also. We call them receptors. Yes, we need receptors to respond to stimuli, to acquire this stimuli, and then to respond to stimuli. So cells possess receptors to hormones, for example, to growth factors and extracellular materials. Yes, without receptors it's very hard to get the stimuli and respond to to the stimuli. The next characteristic is that cells are capable of self-regulation. Yes, so what it means? Cells are robust, that is hearty or durable, some kind of durable, because they are protected from dangerous fluctuations in composition and behavior. Yes, so we have very precise, I mean, regulatory systems that try to somehow keep this balance, yes, inside the cell, keep this balance in chemical composition, for example, like pH or temperature or blood pressure. Yes, all this characteristics must be in a normal range. Yes, that's why we need the constant regulation. Yes, so maintaining a complex ordered state requires constant regulation. So that's why these regulatory systems work very hard to keep this balance inside the cells and inside the whole organism. Yes, and very last characteristic that we must describe, and especially we will describe it on the next Monday, when you also will be involved in this seminar activity, that cells evolve. Yes, this is one of the characteristics. So, of course, the evolution of the first cell is still a big mystery, but evolution of cells can be studied by examining organisms that are alive today. Often quite different cells share many features, including a common genetic code, for example, plasma membrane, presence of ribosomes, and we will see it when we compare now, for example, bacterial cells or plant cells. Yes, we will see that they share some common characteristic like presence of plasma membrane, because we can detect plasma membrane in bacterial cells as well as in plant cells, in animal cells, but also they are different, of course, and this difference is conditioned by this fact that cells evolve. Yes, they evolve in different directions. And this evolution is not simple event of the past, but it's ongoing process. This evolution happens every day, but it needs time, of course, and it continues to modify the properties of cells constantly. For example, one very good example is drug resistance in bacteria. Yes, very good example of evolution of these cells, because time after time, you know that bacterial cells are getting adapted to a new antibiotics. That's why this is a big problem for today in medicine. Yes, big problem, because we have new antibiotics and less effectiveness. Yes, they are less effective time after time. That's why we never use the same antibiotic in the second time if we need it, of course. Yes, we try to use new antibiotics, and this is a very good example of this characteristic that cells evolve. And also we can say something the evolution of this COVID-19, of course. Yes, because we already know many things about the evolution of this viruses in a very short period. Yes, this is very strange, of course, but this is characteristic to viruses, because they can evolve in very short period of time, and that's why we have so many strains of this virus. Yes, this alpha or beta or gamma or delta or like Omicron. Yes, recently we had an Omicron, so this is very good examples of evolution. So now let's start talking about the basic types of the cells. Yes, in reality, we have two basic types of cells, and you know about it already. Some cells belong to eukaryotic cells, some cells belong to prokaryotic cells, and the main difference between these two types of cells is presence of presence or absence of nucleus, of course. Yes, the cells that contain nucleus, we call these cells eukaryotic cells, but these cells that don't contain nucleus, we call them prokaryotic cells. And very good example of prokaryotic cells is the bacterial cell, of course. So not only bacteria, so here we have the green blue algae, or of course, yes, bacteria, green blue algae, and archaebacteria belong to this group, I mean prokaryotes. So what we can say about this, the main characteristics of these cells, we can say that they have a so-called cell wall. Yes, what is cell wall? This is some kind of protective coat outside. Yes, outside layer of that is for protection for this bacteria. But this is some kind of similar characteristic. Because also plant cells contain cell wall, this similar structure. Yes, so please remember that bacterial cells contain cell wall. This is kind of protective coat, and also they contain plasma membrane, and this is similarity, of course, because all these cells contain plasma membrane. It can be eukaryotic or prokaryotic cells, and you can see plasma membrane under this cell wall, of course. Also, they contain cytoplasm and the circular DNA. Yes. So this is some kind of difference, yes, because eukaryotic cells don't contain circular DNA, but bacterial cells contain circular DNA. It means that they have only one molecule of DNA actually, and that's why only one chromosome, if we can call it chromosome, of course, yes. And they have some flagella or cilia outside, and they can move with the help of this flagella and cilia, of course. And one more similarity that we can detect, this is the presence of ribosomes. Yes, I hope that you remember what are the ribosomes. They are very tiny organelles inside the cytoplasm, and the main function of these ribosomes is protein synthesis, yes, or translation. They participate in translation process. They are very little organelles, and they don't contain any membranes. Yes, they are not membranous organelles, but still they belong to cytoplasmic organelles. Yes, and so the presence of ribosomes, we can say that this is similarity between prokaryotic and eukaryotic cells, because also eukaryotic cells, no matter it can be plant cells or animal cells, also contain ribosomes because they also need a protein synthesis. Yes. So you know now how to describe these cells. Yes, you must say something about similarities, and you must say something about differences, of course. Yes. And what is nucleoid? Nucleoid is this place or place in cytoplasm where we have this circular DNA, very simply speaking. So we call it nucleoid because you know that bacterial cells don't contain any nucleus as a distinct structure. Yes, they don't contain nucleus, but they contain nucleoid, the place, some fragment or some part of cytoplasm that contains the DNA, the circular DNA. So now what we can say about plant cells. Plant cells, as I already mentioned, also contain cell wall, as we already detected in bacterial cells. This is quite a rigid structure. It's responsible for the shape of the cell, of course, because it's strong and rigid structure. It's composed of cellulose mostly, and we will talk about it later when we discuss the chemical compounds of the cells. It's built up with the help of cellulose molecules. We have a nucleus, of course, this distinct structure that contains genetic material, of course. We have a nucleolus, we have chromosomes inside, yes, and DNA molecules. We have nuclear envelope, so-called, that protects the genetic material. Yes. Also, in cytoplasm, we can detect some organelles, cytoplasmic organelles like chloroplasts. Yes. First of all, I want to talk about chloroplasts because this is a difference. Yes, of course, no other cells contain chloroplasts. No, and I mean animal cells, for example. Yes, we can't observe chloroplasts in animal cells. We can see them only in plant cells. Yes, chloroplasts that contain chlorophyll, and this is essential to carry out photosynthesis. Yes, of course. You can detect here mitochondria. Yes, and this is similarity because animal cells also contain mitochondria. Somehow we must compare animal cells and plant cells. What are similarities? What are differences? For example, if we take this vacuole, yes, the very big part is occupied by this organelle. This is vacuole, and this is characteristic to plant cells. Yes, for example, not characteristic to animal cells. And also, I want to say something about the centrioles. I hope you remember about centrioles, that this rod-like structures, you see them. The centrioles are characteristic to only animal cells. Yes, I want to somehow to remember. Yes, these organelles are characteristic to animal cells and they are not characteristic to plant cells. Yes, please remember, because you can't find any centrioles in case of plant cells. Yes. And what else? And also we can say that lysosomes is less characteristic to plant cells, because we have another organelle like peroxisomes. Yes, that is more characteristic to plant cells than lysosomes. But also we have other organelles, I mean membrane-bound organelles, that are similarities. Yes, we have detect them in plant cells as well as in animal cells. What kind of organelles they are? They are Golgi complex, for example. Yes, you see this cistern, like very characteristic structure that's characteristic to Golgi bodies, so-called Golgi complex. You can see endoplasmic reticulum. Yes, we have two types of endoplasmic reticulum, rough and smooth. Yes, so rough endoplasmic reticulum always contains ribosomes at the surface, and that's why we call this structure rough endoplasmic reticulum. And another type of ER, this is smooth ER, because it don't contains any ribosomes, and so we have some differences. That's why in functions of these two parts. Yes, for example, if we say that a rough ER is involved in this protein synthesis, yes, we can't say the similar thing regarding the smooth ER, because smooth ER can't be involved in protein synthesis because they don't contain any ribosomes, but they ensure synthesis of lipids, for example, some steroid hormones, for example, some membrane lipids, cholesterol. Yes, so this is the main function of smooth endoplasmic reticulum. So they somehow distribute these functions. Yes, but there are some functions that they share, of course, and we will discuss it later during our lectures, of course. Yes, so as I told you, cell wall is characteristic to plant cells, but not characteristic to animal cells. So this is another difference. We can't find any cell wall inside animal cells. Yes, we only have a plasma membrane, and we only have some additional glycoproteins at the surface that help cell in communications, of course. Yes, because two cells, these cells are in constant communications with another cells, yes, with neighboring cells, and that's why they need these glycoproteins outside, yes, that is connected to plasma membrane, of course, yes. Also, I want to say something about these tables, yes. That's why, as I see, only 1 minute is left. That's why I want to ask you to join again, okay? Maybe 5 or 10 minutes will be needed to somehow finish this lecture, okay? Can you hear me? So join again.
Yes, Doctor.
Okay.